Hi there, and welcome to this video on GCSE Biology for the AQA specification, focusing on monoclonal antibodies and in particular on the uses of monoclonal antibodies. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello, and welcome to tutorial 2 of 2 on monoclonal antibodies. So in today's session, we're going to be having a look at how we can actually use monoclonal antibodies. And if you've watched the last session, what we did there was we introduced the concept of monoclonal antibodies, and then we looked at how they can be made both naturally within the body and artificially in vitro. So in vitro just means out of the body, so in a test tube, for example. So today we'll be looking at the uses of monoclonal antibodies, and so some examples of this are in pregnancy testing, investigating blood, treating cancer, and then also we're going to be having a look at some of the side effects associated with monoclonal antibodies. So let's start off at going over 4 key properties of monoclonal antibodies. Well, they can be used for the following purposes, so pregnancy testing, research testing, lab testing, and treating diseases. So let's just go in to explore this a little further. So pregnancy testing uses monoclonal antibodies to detect for human chorionic gonadotrophin, or HCG for short. So when a woman is pregnant, she releases this hormone, and it's unique to the pregnant woman. So a person who is not pregnant will not have HCG present in their urine. Only a pregnant woman will. And so, therefore, a monoclonal antibody can be used to detect this HCG, which will be found in the urine of the pregnant woman. So let's take a little look at this in more detail. So just keep in mind, throughout this whole presentation actually, we can use fluorescent dyes when doing tests with monoclonal antibodies. And so these fluorescent dyes are going to evoke colour changes that will help us detect whether the test is positive or negative. So this is a common theme that occurs throughout biology, especially within the theme of antibodies. So just watch out for it here. So first of all, the pregnant woman, she has her pregnancy test over here, and she's going to urinate onto the test strip. And if she is pregnant, so we're going to look at the case in which the woman is pregnant, she will have HCG, human chorionic gonadotrophin, present in her urine. So if HCG is present, it's going to bind the free monoclonal antibody. So these here are your monoclonal antibodies. Oopsie. Monoclonal. And I'm just going to write antibodies as ABS for short. So they are specific to HCG, so they're only going to bind HCG and nothing else. So because this woman is pregnant, she's going to have HCG in her urine, and we have monoclonal antibodies that are on this test strip, and they're going to bind the HCG that's in her urine. And these monoclonal antibodies are special because they are actually equipped to change colour. So they have a dye enzyme attached to it. They're not equipped to change colour, but they have a dye enzyme attached to it. So, now at the test site, which is this bit over here, the HCG that is bound to the free monoclonal antibody, and remember this free monoclonal antibody has the dye enzyme attached to it.
So this whole complex is going to enter the test site. So you can imagine that as it moves down here, the urine is here, it's going to move down with the complex of HCG with the monoclonal antibody, which has a dye enzyme attached to it. It's going to move down to this test site, and at the test site, the HCG free antibody complex will bind to another fixed antibody using the HCG. So you can see here, you've got your HCG monoclonal antibody here, bound to the HCG, and it's going to bind to another antibody that's attached to this surface, so it's attached here to this test strip. And this will bring the enzyme, which is attached to this HCG antibody complex, close to a dye substrate. So imagine the dye substrate being here, and this is your HCG, this is your monoclonal antibody that we had at the start, and they've all been brought into a close complex, and this is going to induce a colour change. Therefore, we get a colour change because this complex is going to lead to this colour change because we've got the dye enzyme that's attached here that is being brought into close contact with this, and that's going to induce a colour change. Now, here at the control site, there is a fixed antibody as well. So just like how we had a fixed antibody at the test site, this was our fixed antibody, this pink antibody, we also have a fixed antibody at the control site. But this antibody isn't going to bind to our HCG complex monoclonal antibody. It's going to bind to any free antibody that's lurking around, so any antibody that hasn't bound HCG. And it will also cause a colour change here. Now this might seem a little bit confusing, why are we getting the same colour change? But don't worry too much about that. All I want you to take from this is that there is a control site that is going to be positive, and there is a test site that's going to be positive in the pregnant woman. And that's because of these antibodies binding the fixed antibodies that are on the sites. And this control site is important because it proves the test works, as we will now come to see in the non-pregnant woman. So if a person is not pregnant, they're not going to have any HCG lurking about in their urine. So therefore, when the pregnant woman urinates onto the test strip, we're just going to have lots of free antibody that's present on the test strip that's not binding to any HCG because there is no HCG present here. Therefore, the free antibody will move through the test site, and so it's going to move in this direction, this free antibody. It's going to move down here. There's going to be no binding in the test site. Okay? Now, why is that? Well, we don't have this complex being formed because we don't have any HCG. So how is it supposed to bind to these fixed antibodies in the test site? Well, it can't. So that means that the free antibody is going to pass down, there's going to be no binding here, and it's going to go straight to the control site. But hang on a sec, take a look at, compare these two diagrams. This scenario can happen. So, in the control site, the free antibody is going to bind the fixed antibody, and there's going to be a colour change here. So, note the difference between these two diagrams. If a person is pregnant, the HCG-free antibody complex will form in the reaction site, and this complex binds in the test site, causing a colour change here. But at the same time, there will be some residual free antibody, which is just simply not going to bind to the HCG, perhaps because it hasn't had the lucky chance to come into close contact with it. So there's still going to be a little bit of free antibody left over, and it's also going to induce this colour change in the control site. So we're getting two lines here in the pregnant woman. Whereas in the non-pregnant woman, we're not going to get any positive test in the test site because there is no HCG present in the urine. So all the free antibodies are just going to continue onto the control site and we're going to get a positive test here. And this just proves that the test works. So that's the purpose of the control here. So this is a really, really useful diagram, and if this didn't quite go in the first time you listen to it, don't worry at all. It is a little bit complicated when you first come to it, but just rewind the video and go back and listen to those last 5 minutes again, and perhaps draw out this diagram yourself and annotate it in terms that make sense to you. So just to summarise what I've just said, in a positive pregnancy test, so where the woman is pregnant and there is HCG present in her urine, you're going to get HCG binding to the free antibody, which is located on the test strip. And the bound HCG antibody complex binds to the fixed antibody on the test strip. And we're going to get two colour changes happening, one in the test site and one in the control site. However, in a negative pregnancy test, the woman is not pregnant, she doesn't have any HCG present in her urine, so we're going to get no binding to the free antibody. That means we're going to have lots and lots of free antibody in the urine, and nothing binds to the fixed antibody. The only colour change that happens is in the control site, so the free antibody is going to be binding to the fixed antibody in the control site. So, as I've just said, if a person is not pregnant, the free antibody moves straight through the test site, straight through to the control site, so you only get one band forming in the non-pregnant woman. But if a person is pregnant, the HCG free antibody complex, and we call it, this is a little bit confusing here, it confused me at first, I'm not going to lie. This part is the HCG that we were talking about in the urine, and then by free antibody we just mean the antibody that was located on the strip. So it's not HCG free, it's the HCG bound to the free antibody, and that together forms the complex. And so if the person is pregnant, the HCG free antibody complex will form in the reaction site as well as in the control site, so we're going to get two bands. So this is just summarising it in a different way. So we're going to be getting the urination onto the reaction site where the free antibody in solution traps the HCG, and the free antibody, remember, is attached to a dye enzyme. This is then going to move along the test strip, and the antibody that's bound to the HCG is going to bind to the fixed antibody here, which is located on the test site. So we're going to get binding of our free antibody HCG complex. I'm just showing the HCG as this purple dot there. That's your antibody, whereas this is your fixed antibody, which is located on the test site. And then, so that reaction is going to occur in the pregnant woman, but then also we're going to get the movement of any free antibody that hasn't bound HCG just by chance, and that's also going to bind here. Or actually, I could have drawn this a lot better. If I just go back, I should have really drawn your HCG in there. So that's your HCG, that's your antibody. Whereas here we're going to be getting our free antibody with no HCG attached. Okay? So in the pregnant woman we're going to get a colour change in the test site and a colour change in the control site. In the non-pregnant woman we're not going to get a colour change in the test site, only in the control site, because we're only going to have these free antibodies in the solution. We're not going to have any antibody bound to HCG. So 2 lines is a positive result. Now, as well as using monoclonal antibodies in pregnancy tests, we can also use them in investigating blood. So to detect whether particular antigens or antibodies in patients' samples are present or not. And these are known as enzyme-linked immunosorbent assays, or ELISAs, and they work in a very similar way to the pregnancy test because they use monoclonal antibodies that bind to the antigen present that we want to detect, and this leads to a subsequent colour change reaction due to an enzyme. So let's just look at this in a diagrammatic form. So we have an antigen that's immobilised on this plate here. Then we're going to incubate our test serum with that, and any antibodies that are specific to our antigen are going to bind our antigen. We're then going to introduce a second antibody into the solution, and this second antibody is labelled with an enzyme. Here, this yellow blob is your enzyme. And so we're going to incubate this whole thing together with our secondary antibody with the enzyme attached to the secondary antibody. We're then going to wash all of that, so we're going to remove any unbound. When we wash, we're going to remove any unbound secondary antibody. And then what we're going to do is we're going to add a label, which is then going to introduce a colour change when the substrate is added. So when we add our substrate, a colour change will be induced, and this will show that we have this binding scenario happening. So we can use ELISAs to measure the levels of hormones in the blood, and also to detect presence of specific pathogens in the blood. So in the same way, when we say measure the levels of hormones in the blood, monoclonal antibodies can be made that are complementary to certain hormones. So this is kind of how the pregnancy test worked as well. We had a monoclonal antibody that was complementary to HCG in the urine. And in terms of detecting pathogens, monoclonal antibodies can be made that are specific to a particular pathogen and can be used to identify the presence of this pathogen within the blood. So now let's look at how monoclonal antibodies can be used in treatment. So cancer cells often have antigens called tumour markers, and because we know of these tumour markers, we can design monoclonal antibodies that are specific to these tumour markers. So these monoclonal antibodies, when they're injected into the patient, they're going to go and find these tumour markers and they're going to bind them. Now, why would we want to do this? Well, we can do clever things. We can attach drugs to the monoclonal antibody, and these drugs can then be carried to the cancer site directly. Or alternatively, we might want to attach radioactivity to the monoclonal antibody, and so when the monoclonal antibody binds its antigen on the tumour, we can then identify the location of the tumour in a scan. Or we can attach a chemical to the monoclonal antibody, and this can be then delivered to the cancer cell, and this may inhibit the growth of the cancer cell or kill it. So I think it's much easier to think of this in a diagrammatic form. So for example, with example number 1, we've got our antibody carrying an anti-cancer drug. So this is our antibody. You can see we've attached it to the anti-cancer drug, and our antibody is specific to, let's say, this pink triangle over here. So it's going to be looking for it, and it's found it on this cancer cell. The monoclonal antibody binds, and then we're going to get delivery of the drug to the cancer cell. In a similar way, we have this monoclonal antibody that's attached to radioactive material, and it's going to bind this cancer cell, and therefore the radioactive material is now going to be bound to the cancer cell, and this may show up on a scan, which will be really useful in identifying whereabouts the tumour is in the patient's body. Or alternatively, the antibody may carry a chemical, And this chemical may do a number of things, and an example of this is it may attract immune cells. Excuse me. And as we learned in the tutorials on communicable diseases, immune cells are very good at launching attacks against foreign bodies, and so if our monoclonal antibody is attracting immune cells to the cancer site, this could be useful in launching an immune attack against this harmful cancer cell. So this is also a really, really good diagram to draw out in your notes and annotate on. So let's have a look at some of the side effects of monoclonal antibodies. Well, just like all drugs, patients can develop allergic reactions to monoclonal antibodies, so this is one negative. Also, monoclonal antibodies have been known to cause deaths in some patients with multiple sclerosis, which is a type of disease. Also, monoclonal antibodies rely on detection of a specific antigen, but remember, antigens can vary and change over time. They're not always constant because, for example, a gene mutation may change an antigen, and if the antigen changes slightly, the monoclonal antibody may no longer recognise the antigen, and therefore it's useless. Also, we use mice for the production of monoclonal antibodies, and so there are concerns over animal rights with this process. But of course, there are lots of positives associated with monoclonal antibodies. They can be used to treat many diseases and can be used to detect early pregnancies to help mothers. Also, monoclonal antibodies as cancer treatment yield fewer side effects than alternative therapy. And also, it's a very quick reaction as the monoclonal antibody just has to bind the antigen and then we're going to see a colour change and we can monitor this colour change. So therefore, monoclonal antibodies are really useful in diagnosing diseases really quickly, or not even diseases, also diagnosing pregnancy. So we really have covered a lot today, so I'd say it's a good idea to go back, get those diagrams down in your notes along with any annotations that are helpful for you. So thank you very much, well done for today, and I'll see you for the next session.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.